are still coming. Yeah, since all people slowly come in, I will say welcome to everybody now on the screen or in the room already, and to everybody who is coming. We continue our lecture on the archaeology of the Caucasus, and I will host this day's meeting. And I have the very big pleasure to introduce Wolfgang Haak and Ayşin Galici as the speakers of today. They will tell us very interesting, I hope so, news from a site of Caucasian archaeology, which is rather new and in which, in fact, Wolfgang and Ayşin and some other colleagues have, let's say, developed only within the last several years. This is paleogenetics, and the topic of today will be the paleogenetic of the wider Caucasus, presented by Wolfgang Haak, and with an summary in Azeri-Iranian by Ayşin Galici. Wolfgang Haak, I think, is, I don't have to introduce him too much. He has maybe the most cited prehistoric article written, which is on the market at the moment. Wolfgang is a paleogeneticist, and he worked for a long time in Mainz at the group of Kurt Alt and Joachim Burger in the wider field of then anthropology, physical anthropology, and bioarchaeological aspects. He defended his PhD in Mainz, worked there also as a postdoc, and then went for the Australian Centre for Ancient DNA Studies to join the National Geographic Ancient Genome Program. That's on the other side of the globe, but he returned, thank God for us, in 2015. He returned to the Max Planck Institute for the History of Mankind in Jena, headed by Johannes Krause, and was there a group leader with his own group. And that's also where we first met on topics of paleogenomics. And since this year, since 2021, his group has shifted to the Max Planck Institute for the Wolfgang, help me.
Evolutionary anthropology.
Evolutionary anthropology. It's too fresh now. And so now he and his group has moved to Leipzig. He has rewarded, amongst others, a big ERC grant, PaleoRider, and in this ERC grant, Ayşin Galici is a PhD student. She joined the group in 2018 and is also now working on paleogenomics of the North Caucasus. She is a PhD student, as I said, and initially from the University of Ankara. Of course, the genetics of the Caucasus have also some other groups seen, and I think Wolfgang and Ayşin will report on that. And with this, I will hand over the microphone to Wolfgang, and we will hear later Ayşin's summary.
Yeah, thanks so much. Hello to everyone. It's a great pleasure to be here. Thanks for having me in this lecture series, and thanks to Sabine for the nice introduction. I'll start sharing my screen. Let's see. So the presentation is here, and now should see this. And now you have to let me know or give a sign whether you can see the full screen or the presenter mode. Looks good.
Yeah.
Yes, everyone can see and hear me. That's great. And it's recording. Good. Yeah, thanks again for having me. You should see a kurgan in the distance. That's correct. You're in the right topic. I will talk about the genomic history of the wider Caucasus region. And the motivation for this work that started a couple of years ago when I came back to join the Max Planck Institute for the Science of Human History was that we came together as a group with colleagues from the DAI, amongst which were Sabine Reinhold and others from the Eurasia department, but which had already worked intensively for several years in the region and beyond. So that was a great enterprise to shoehorse in. And for us, the motivation to work in this region generally stems from the Caucasus being such a key geographic region that is bridging Europe and Asia and the Middle East. So it's, of course, this in the center here, we see this high mountainous region that is bounded by the Black Sea to the left and the Caspian Sea to the right, and then connects the Armenian and Iranian plateaus and parts of Eastern Anatolia with the Fertile Crescent, as we know it from archaeology. And then north of it, it abuts the endless grasslands of the Eurasian steppes, which have also played a great role for Western Eurasian history. And then as a consequence from that being a geographic feature, and because perhaps occasionally also a refugium or a sink region, it certainly harbors then as a consequence this fantastic or substantial cultural and linguistic diversity, as we see on the left-hand side. And if we think back really in time and take, for instance, the Tbilisi finds or the Neanderthal finds in the Mesmaiskaya cave into account, it was potentially inhabited by hominins since times immemorial. The diversity in the region can also be seen in the archaeology itself, as shown here with the chronology of archaeological cultures or phenomena or complexes that are broadly sorted by geographic regions here in columns. And these are tied to the equivalent analogous chronologies of the Southern Caucasus here, for instance, Eastern Anatolia, and also Northern Mesopotamia, that is linking everything across the main mountain range. It certainly also mirrors the myriads of influences that have reached the Caucasus from outside, but also as a source region and a sphere of influence that then reached the neighboring regions in reverse, and that at various points in time. So things come in and go out, and this is highly dynamic and diverse. The geographic diversification is obviously reflected in cultural phenomena that are sometimes more associated with mountainous groups or hilly groups like the Maikop or the North Caucasus culture here that we will talk about. But then on the other hand, there are also groups that we can find in the abutting steppe zones that adhere to a more steppe economy or a steppe way of life, such as the ones that are well known as the Yamnaya pastoralists or the catacomb associated groups. Given the complex history of the region that is, of course, attested in archaeology, as I showed, but also the cultural and linguistic diversity observed today, we wanted to explore this region genetically and also add a temporal dimension by which we were attempting to do that by studying the ancient human DNA from select prehistoric individuals from associated cultures that we had just seen on the chronology. And in particular, from this ancient DNA perspective, we were interested in the following questions. When and how did the genetic diversity that we observe today form? And then based on the 2015 paper where we looked at the Bronze Age formation of the genetic diversity of Europe, we also saw that there was an influence coming from the Eurasian steppes, and we wanted to also then retrace where is actually this steppe ancestry coming from. And we We knew from this study that the steppe ancestry was formed by, on one hand, northeastern European hunter-gatherers, but the other counterpart that was adding to the equation was somewhere located in the Caucasus. And by that time, we only had modern-day Armenians as the best proxy to bring together what ultimately had formed in the past, the steppe ancestry signature. And of course, then once that is sorted, we would think that we could look for gene flow from the Caucasus during times when the exploitation of the resources of the steppe environment versus the mountainous environment was intensified. And also in the greater scheme of things, understand the means by which Europe, the steppe itself, but then also the earliest urban centers in the Near East, which certainly predated everything that we had happening here, were then connected and spread their influences to the surrounding regions. Yeah, I forgot to add this. Before we delve into the genetic results, here's a quick overview over the general gene flow. Not gene flow, workflow. That was a Freudian geneticist's mistake. So the general workflow in ancient DNA labs and the various critical steps that are needed to also ensure good data quality, to make sure that what we're talking about is authentic. That is important, that we're not talking about false positives, and everything is actually coming from really precious and highly valuable prehistoric samples that are coming from well-contextualized archaeological background. So when those samples here enter our lab, we prepare them. There are a couple of biochemical steps with which we then dissolve the bone powder that we drill out from particular samples. And this is all happening in dedicated clean rooms where we prevent modern day contamination to have an effect on these samples. And then once we extract ancient or DNA molecules from these samples, we immortalize them through the generation of so-called DNA libraries, which is a synthetic amplification of every molecule that was extracted here to the point where it can be stored for, eternity is essentially. And a part of that is then usually sequenced. So we read out the genetic information of those molecules. That goes into a step of bioinformatics and quality control to ensure that what we're dealing with is in fact authentic and ancient. And then we also want to know, is there actually anything in there that is human, that is of interest for us? And if that is the case, and it's enough, then we can always sequence more and produce what in the end amasses to an entire genome. And that's a lot of information that we can work with in comparative population genetic analysis. If it's not enough, then we have a little trick, and that is the targeted enrichment of informative regions in the human genome, and by which we fish out those regions directly with a DNA hybridization method. And that then captures the regions of interest, which then cost effectively get sequenced again, and then it undergoes another round of bioinformatics followed on by comparative population genetics. But we can also look at individual based information such as kinship or biological relatedness, I should say, but also things like selection in larger cohorts and so on. Now over to the ancient DNA data itself. In 2015, my postdoc fellow Zhao Wang and I joined the colleagues from the German Archaeological Institute and the colleagues in Russia to sample teeth and bones from the North Caucasus region. Here's a map showing the geographic locations of the sites we had sampled from. You'll note that the most collection points are from the North Caucasus, some in Georgia, and Armenia as well. In population genetics, then, we have to do some preparation work. We often use multivariate statistics to break down the complexity of the data that is needed, and we also need to prepare the data that is used for comparison. Because if you just have one data point that is naked in space and time, it doesn't help us anything. Compare it to archaeology. If you just had one pot from prehistory, you wouldn't be able to locate it and put it into its place. So you need always your vocabulary and your typology essentially, and understand that in order to place things in the past. We often use multivariate statistics such as principal component analysis to then reduce the complexity, the dimensions of the genome data, because we now produce variants in the ranges of hundreds of thousands that differ between populations and individuals of various populations. And in order to master or get on top of this diversity, we use those statistics to break that down into dimensions that we can handle and that are also informative in the end. Here you see a classical principal component plot in which we have 84 West Eurasian populations. You see immediately that the genetic diversity between the groups, or each dot here is an individual, is broadly mirroring geography. So you can follow here the descriptors. And you also see that, at least in modern day populations, there is a bit of a distinction between those ones north of the Caucasus, the main mountain range, and the ones south of it. The ones south of it are linked intricately with Levant and the Near East, and this would be Southeastern Europe, and then we go into Northeastern Europe, and that would be Southeastern. That's the Iberian Peninsula. This is the Italian boot by Sardinia. So keep that in mind. As the modern day genetic situation, because this is now grayed out as the scaffold in the back, and then we want to project ancient DNA data onto it. This is relevant published data that highlights the important clients of ancestry that we and others have described to reconstruct the genetic history of Western Eurasia in general. They are clients of admixture between preceding hunter-gatherer groups, such as the ones in Europe's west, the ones in Europe's east. There are hunter-gatherers from the Caucasus and Iranian Neolithic individuals here. The Anatolian Neolithic individuals are down here, and Levantine Neolithic individuals are also down here. And you see that over time, these things mix and mingle, and the diversity in the past did look not a lot like the one that we observe today. So it hasn't been the case that the geography and the differences that we see in populations today was formed after the first peopling events of the regions, and has been kept in place ever since. No, there was a lot of dynamicism, and you can glean already from this picture here that distinguishes maybe the situation 8,000 years ago with present day, that a lot has happened to shuffle people around and mix and merge them into new types of ancestries. So now a brief disclaimer, of course, while we're at it, we have to talk about terminology and the way in which we use it. It is absolutely clear that for the sake or the purpose of such a presentation, we can only use relatively broad descriptive categories that can be subjective and ambiguous in their attempt to convey additional contextual evidence. So you have to allow that. We have to simplify things. And when I say Anatolian Neolithic, it means that we talk about a genetic ancestry cluster that is shared between largely contemporaneous individuals that are associated with a predominantly agricultural subsistence, and also broadly assigned to a respective archaeological time period, here the Neolithic, and then also with the geography term, then from a particular geographic region. And we could, of course, also keep it simple and just talk about population 1 or population A, but then you would simply lose a lot of track, lose track soon enough. And people have thought long and hard about what would be the best way to go forward. There's now an opinion paper on that, but of course we can refine these as we go along. Now, I kept you distracted from the actual going to the bottom of it all. Here now are finally the newly typed ancient individuals in symbols here with black outlines. The first thing you notice that this new data from the North Caucasus, where we collected, does form two relatively distinct clusters or data clouds, one over here and one over here. And the colors generally here are as follows. Orange is what is maximized in Anatolian Neolithic, blue is what is maximized in European hunter-gatherer individuals, and green for everything that is maximized in the Caucasus and the Iran. As is on the PCA, we also see it in admixture. That is another way to break down ancestry per individual. Here on the y-axis, you see all the individuals that we had added to it. And yeah, if we sort them by ancestry, it also follows essentially those two different data clouds. This is what we have here. And what we do on the x-axis is that we sort them through time. And now we see that we can broadly describe, and this is what we did here, a group that we call tentatively steppe, and another group that we call tentatively Caucasus or Piedmont or mountainous group. And you see that individuals from each of these clouds do share ancestry that is relatively similar, and that stays relatively similar throughout time. Some of them are as old as older than 6,000 years, and some of them are ending in the late Bronze Age, and the same time transect is also observed in the steppe. And what is also interesting is that we can also look at the mitochondrial DNA, so the uniparentally inherited markers, such as the mitochondrial DNA or the Y chromosome, which is the paternal equivalent on the father side. If we look at the father side, we see a diversity that is mutually exclusive. So what you have in the steppe, you don't have in the Caucasus group, and vice versa, whereas the situation is different and quite homogeneous and similar on the maternal side, where we have a slight frequency differences, but this is very comparable and suggests there's a semi-permeability of the two regions, which is accompanied by a relatively substantial sex bias. So not everyone can move, but you can move between the regions. Speaking of regions, when we map the genetic data back onto a geographic map, we actually see that this genetic pattern that we see is also geographically confirmed. Not only because we named these things afterwards, those clusters, after mapping it onto the map, onto the geographic map. We also see a relatively clear demarcation line. This is the gray dashed line here between the Piedmont, so the flanky northern hills of the Greater Caucasus, and then the real steppe, which is the endless flat with the endless horizon to the north of it. And furthermore, if we then look a bit more into the ecogeographic zones, we see another subtle signal, and that is that these individuals that are north of this blue line are actually carrying a slightly different ancestry. So they have a bit more additional components here, such as this mint and red ancestry component here that we only find here in the more drier and saltier steppes of the northeast here. And that had us quite intrigued. But first we wanted to also integrate the data with the archaeology. And here we make further interesting observations. For instance, individuals that are associated with the archaeological mycop culture here were found in both groups, in both data clouds, as in the Piedmont region, as well as in the steppe to the north. And this is why subsequently we distinguished mycop here, or mycop proper, in this group, in the Caucasus group, from the steppe mycop here in the steppe group. Classically, of course, the mycop horizon is located in the northwest Caucasus, so those red dots here. And we do in fact see a highly similar material culture in the burial practices between those in red and those in light blue, which are associated with a more steppe way of life. So they that's interesting, so that they say share cultural traits and practices. However, steppe mycop and mycop proper do genetically differ substantially. And you also notice that they're to some degree derived from their local preceding Neolithic ancestral populations with which they share a lot of ancestry. And this is what we want to look at in future in more detail, how that actually happened or came about. Looking at other contextual data, we can also observe that both mycop groups had a different economy and likely exploited different ecological zones with minimal overlap, for example, of livestock or by pastoral movements. And this is tangible here from the isotope data, where you see that both the humans and the animals occupy a different isotope space here in the dietary isotopes, and there's hardly any overlap between the groups here. So there are substantial differences that are akin to the genetic differences observed as well, despite the cultural similarities. So the mycop, steppe mycop version has had us intrigued and urged us to explore this phenomenon a little further, in particular the subtle ancestry signals that we saw, this mint green and the red. And that one we observed in quite characteristic individuals, which we like this one, which we effectively call Mr. Sharach al-Sun. This is a relatively robust male individual that was buried sitting on his wagon. He had 20 or so healed fractures and probably was one of the first race car drivers of his time. So his ancestry and that of the other steppe ancestry individuals needed further exploration, and in order to do so and place it on a wider global scale in comparison was needed, and therefore we extended the PCA to include other Eurasian populations as far as the northeast and southeast of Asia, and we see that the steppe individuals or steppe mycop individuals follow a cline that is ultimately linked to ancestral North Eurasians, such as the 20,000-year-old Malta boy or individuals from Afontovagora, those individuals here in black. So there's an ultimate ancient genetic link, and those individuals here that are so ancient also trace a bit of this red component. This red component is ultimately maximized in today's Nganasan and is carried also by many other people that are Uralic speaking today. So that is an interesting observation. What we did not find among any of these was the mint green proportion. So we had to look even further and extend the global PCA. And by adding Native American individuals, bingo, we had found a link, and that is what is visible here. In all Native American individuals today, we also see shared ancestry that is here maximized with steppe mycop individuals, which is ultimately linked through those ancestral North Eurasians here on this admixture cline, which is quite surprising, or was surprising to us at the time. Today, we know that we don't have to go quite as far as to the Americas to find this ancestry. We also find it east of the Urals in West Siberian hunter-gatherers. So this is very likely that it was maintained there or persisted until more recent times, or at least recent prehistoric times, where it also provides a geographic link to that region potentially. That is quite intriguing to us, but also highly interesting to see that there's quite a diversity also in the steppe cloud here. We also find, and that is even more intriguing, evidence for admixture between two individuals in the form of those two outlier individuals here. I should highlight them. They are also from a steppe mycop context. You see them here, but they carry additional ancestry that is also found in the Caucasus cluster. And as no surprise, they also shifted more in that direction. While this might not account for a complete transfer of the material culture from mycop proper to the steppe, at least it shows that the two groups were a little more than culturally connected, if we want to say so. And we also find subtle hints for another episode of gene flow in the otherwise relatively stable genetic continuity on both sides in these clouds. And the other one is seen in the Middle Bronze Age, late North Caucasus culture, which also shows subtle traces. And these are those individuals highlighted with the blue circle. These are coming from a site Kabardinka in the Piedmont. Another obvious feature on the PCA is the observation that if we took the Neolithic here, so the oldest samples as a local stratum, we do notice a subtle shift of everything that follows in the early and Middle Bronze Age, including the Yamnaya and Catacomb individuals. That these are slightly shifted towards the Middle Neolithic farming groups of Eastern Europe, such as global amphora, but also the Tripilia and Cucutane individuals. Archaeologically, this is also not too far-fetched, given that the steppe groups are budding late farming cultures in Eastern Europe and the regions of today's Ukraine and Moldova, as well as in Romania. So we tried to model that. And in fact, it's clear that, and this is a bit technical here, but if we use finicky ancestry modeling, we can at least say if one of the source populations or the populations that we want to test requires several ancestry sources, or whether it's happy with just one ancestral group giving rise to a succeeding group. And here, what it tells us, at least for everything in the steppe, that it requires a bit of everything. So most of them, starting with the Yamnaya Samara, also require a bit of Western European hunter-gatherer, but also an Anatolian Neolithic ancestry, meaning that there must have been contact with the farming world. Archaeologically, there are also a couple of subtle hints, such as hints as to the interaction between farming and pastoralist groups. Already in the 4th millennium, there are similarities in these stone cysts, for instance, between one in the late Neolithic Bernburg culture in Görlitz in Germany, but then there's also one that looks very similar, especially if you stay in front of both of them once in Novosvobodnaya Kladi in the North Caucasus. This is also very exciting to see those similarities and see subtle hints of the connections here too. Another important observation that we could make in these North Caucasus kurgans is the one, or the fact that those burial mounds as a whole can serve as micro transects through time. For instance, here in the Mount of Marinskaya 5, we can demonstrate at a local level the shifting cultural and biological affinities of the community which use this place, or the communities which use this place for burials over time. The first individual here, pointed out, is an early steppe mycop associated individual up here in the steppe cloud in the mount with classical early mycop materials such as daggers. Then comes a sequence of 3 individuals of late mycop burials with the correct genetic composition for a mycop individual. 2 of them are in fact second-degree relatives, and the latest graves also with a couple of Bacrania and nose rings still in situ, as here on the right. And then the last grave out here belongs to an individual associated with the North Caucasian culture, which also carries steppe-related ancestry. But interestingly, it preserves the cultural tradition of cattle head offerings here, the green one, and also the high quality metallurgy of the mycop tradition. So you see the dynamics and the going back and forth of the of the genetic ancestry, which is all buried in one kurgan, and attests to the genetic borders that have been shifting north and south during the time, or different genetic backgrounds that have been crisscrossing over the landscape at various times in the past. So I want to summarize that, the first part of it. So we have a time transect covering 3,000 years of prehistory. We have genetic differentiation between groups of the mountains and the bordering steppes. We have genetic clusters that correspond to ecogeographic zones. The Caucasus Mountains, as we can show, were not a barrier to human movement. So we have South Caucasus ancestry north of the Caucasus. We also, despite that, have genetic continuity through time within each of the genetic clusters, but the mountains are not the border is north of it. Yet we have occasional gene flow between the two, as I could show with the two or four outliers. We also have with the steppe microbe ancestry, we have the westernmost and youngest, when we take the Lola individual legacy of ancestral North Eurasian ancestry that connects Caucasus populations with Western Siberia and even further with Native Americans. We also see that with the later early Middle Bronze Age pastoralist, we see evidence for subtle farming ancestry creeping in, and that is prior to the major expansion in the 3rd millennium. Last but not least, we saw that kurgans can serve as micro-regional time transects and showing shifting borders of genetic ancestry through time. So with that goes out a heartfelt thank you to all colleagues that have been involved and all the collaborators that contributed to the study. If you're interested to read up on more detail, in more details, please check out Wang et al. And Nature Communications. It's all open access. And I want to quickly move on to the southern side of the story, where the situation is quite a bit different. Here we have much more ancient DNA data published already. In the North Caucasus, ours was a pioneering study. Here's a lot more going on, a lot more interest, perhaps. But I can by no means also show the complete record yet. I can cherry pick 2 studies in which I was directly involved and therefore know a little more about it. The first one is by a postdoc, Michael Feldmann, now working in Tübingen, that reports late Pleistocene data from Anatolia and the Levant, and from here, and which shows that both regions, at least, so the ancient Anatolia, as well as the South Caucasus and the ancient Levant, were regions that were quite, or harboring quite different genetic ancestry prior to the actual event of farming practices, as you see from the PCA plot there in their respective clouds. And unlike in other regions, away from the origins of the onset of agriculture and animal husbandry, here we do see a continuation of the local ancestry during the Neolithic transition. Importantly, we have here aceramic farmers and ceramic farmers, but also Anatolian hunter-gatherers to compare, but they all fall within the same cluster here. That is quite different to other places such as Europe, where we clearly have then an expansion of early farmers that outcompete in a way local hunter-gatherers. That is not the case here. It's a totally different story in these regions here. And then in a recent study from last year, we continued this time transect from the Neolithic, the late Neolithic to the Bronze Age, in particular in central Anatolia, the northern Levant here, from which we have additional samples, and also some more from the southern Caucasus, like from Azerbaijan. Here's an overview, a map which depicts the several sites from each region, which ultimately also result in little or small regional time transects for each. And all of these range from, as I said, the late Neolithic, nearly 8,000 years ago, to the late Bronze Age, around 1200 BC. BCE. In this study specifically, we added 110 data points from individuals from the Near East, from that time range. And you see them here depicted in the principal component space. You're now probably an expert in reading these plots, but that's the way in which the genetic history is probably best explained. All the new individuals fall broadly within this region that was previously described. So you have the prehistoric Anatolian Neolithic individuals here. This is our Levantine cluster here, and here we would go across to the Iranian plateau here along the Zagros Mountains. This is the South Caucasus also genetically, and this is what we talked about before. This is the steppe cloud. And the data with the black outlines is the new, and that is quite interesting if we walk through that in a temporal pace and say, okay, let's first look at the late Neolithic. We said before in this study by Feldman et al. That the regions were genetically quite distinct, but now we see that this is no longer the case, or that things are from this cluster, and this cluster move away and almost move in data space towards each other, which is indicating a homogenization effect, which is also seen when we model the ancestry, that each of those regions or individuals from each of those regions also carry reciprocal ancestry from the other regions, which means that there's some sort of melting pot forming over time, which is already happening by the time of the late Neolithic, when we take individuals from here, South Caucasus, Tel Kourou in the northern Levant, but also central Anatolia here. And despite the fact that they do still harbor a geographic signal, they're much more closer to each other than the previous early Neolithic or hunter-gatherer groups from each respective region. That is the first observation. And if we then follow on, it looks like the consecutive period after, that's at least 2,000 years that have followed. There is a process, or was a process in place, that led to an almost complete amalgamation of the genetic ancestries from the formerly clearly distinct groups, and irrespective of where you sample from. So we see nearly the same signal, whether we sample from Chamdebel Talasi or Aslan Tepe or Alkhan Tepe, which are all in here, but also Ikis Tepe is combined in this ancestry cloud here. And this despite the fact that each of these sites and regions are several hundreds of kilometers apart. That's the second observation, and we think that it is certainly due to the ensuing millennia, so between the 6th and the 3rd millennium, that were characterized by, we can say, multiple and repeatedly overlapping spheres of influences and expansions out of core cultural centers, such as the one depicted on these maps, like Halaf, Shumotepe, Shulaveri, or the Ubaid core regions. And later on, there would be examples from the Uro core area and Kura Araxes. So everyone contributed at a certain time to everything, so to speak, and all of them, not surprisingly to archaeology, I would think, we're somehow connected through maybe northern Mesopotamia. We can talk about local or temporal hubs at each of those time periods that have consequently added to this homogenization of the gene pool in the Near East over time. We were, of course, particularly interested in finding out when this mixing happened, and there's a particular method that is aptly called dates, with which we can estimate when admixture took place between potential source groups that contributed and those target populations that we have in the center here, which are of particular interest to us. And then we asked, so if we assume mixing of these groups up here and mixing of groups here to lead to a homogenization of the population here. When did this roughly happen? And we repeated this multiple times for these groups here. Then the estimate for this time point, when it happened, is broadly overlapping and tells us that it must have happened roughly 6,500 years BCE, and maybe slightly earlier for some cases in central and north central Anatolia here. So that's that explains what has happened in the time period in between here. And this, despite these overwhelming homogenization effects, there's still a couple of subtle differences between regions that we can tease out if we zoom in, but these are really a small scale and subtle differences that allowed us to detect gene flow, for instance, from neighboring populations here, especially in the northern Levant during the 3rd millennium here, where we see that there's a bit of an genetic affinity that is then increasing over time, especially when we look in at particular populations of interest. These genetic changes in the northern Levant are at the most striking when we look at the PCA plot here. And if we then simply want to model that as these individuals from Alalach and Ebla as a simple two-way mixture between, let's say, the earlier population here from Tel Kordu and anything from up north here, then it's not a simple two-factor or two-source equation at all. We see that we require a bit more Iranian ancestry for those individuals or those two sites here. So everything else is on this cline from central Anatolia to Iran here. This is a big ancestry mixture cline, and the Levant itself is down here, is also contributing to this big mix, but those individuals here are more on the Iranian side, and we hypothesize that this has to do that we possibly track ancestry from the yet unsampled region of northern Mesopotamia. Of course, why this ends up here could be maybe gleaned from the territorial state formation during the Middle and Late Bronze Age, but of course further sampling from ancient northern Mesopotamia or Mesopotamia itself is needed to confirm this idea, but the spheres of influences would suggest that this is a valid hypothesis, and we'll have to keep posted to see what's going to come out of it and whether this can be verified or falsified. Another nice example here from Alalach, in fact, is that we also not only see this shift compared to the earlier Tel Kordu and a potential link to northern Mesopotamia, potential hypothetical link to northern Mesopotamia, but we also have, in the form of this outlier individual from Alalach, an example of, let's call it, individual life history. Here we have a sample taken from quite a tragic event. This is from a skeleton that was found not disarticulated, but in a strange position at the bottom of an abandoned well. And the physical anthropology shows signs for various heel traumas, and the genetic profile shows that it's not a member of the local community, but looks like that it can be traced to somewhere in the Turan BMAC region, perhaps, or somewhere in Central Asia, definitely with a higher proportion of Neolithic Iranian ancestry. So what it effectively means is subject to speculation, of course, or your interpretation. Let's say it could be individual mobility, but it nevertheless showcases the long distance connections to other regions as well, has brought in further not only cultural influences, but certainly also genetic influences, which still to this date add to the genetic homogenization to a certain degree. And with that, I want to summarize, that's in time. What we can say based on the two exemplary or three exemplary studies from the north and the south, and now from the south, we can say that individuals from Western Anatolia, the Levant, and regions in today's Iran are definitely genetically distinguishable during the Neolithic, and is attesting to local genetic continuity and times before. But then from the late Neolithic onwards, in particular, when we estimate this admixture date at around 6,500 years BCE, this is, we observe this gradient of admixture between all regions to a various extent, to the point that about 2,000 years later, it's all homogenized. And then from this point onwards, we see a relative genetic continuity, maybe despite or maybe just due to larger scale political transformations that are happening at the time, which certainly helped amalgamating the genetic gene pool. We do see, of course, also subtle levels of gene flow in the early Bronze Age, especially in the northern Levant. That is likely not from central or eastern Anatolia. As we said, there must be something else is coming in from a little further east, and we hypothesize that we get a hint for a glimpse of what is unsampled but could be ancient Mesopotamia. And the last one was an example of individual mobility in the case of the Lady of the Well in Alalach. And with that, I want to thank all collaborators and co-authors here. This is the main lead author, Irini Skurtaniotti, and a lot of names are on here that might sound familiar to you. Certainly one of the names here. And I want to thank everyone involved in that. That was a beautiful journey. In both cases, and I want to thank again for the invitation and you for your patience, and I'm happy to take questions. But first, I want to pass on to Aishin, who will give you a summary in her language now, and yeah, looking forward to that. Thanks so much.
Okay, should I start? Do you hear me?
Yes, you can. Nice.
Öncelikle Arho'ya ve Sabine'ye məni buraya çağırdıqları üçün təşəkkür edirəm. Və indi mən səy eləyəcəyəm ki, bu gözəl danışmanın Azəricədə bir xülasəsini yapam. Bizim Qafqazda çalışmaq əsasələrimizdən biri bu rayonun Avropa, Asiya, Yaxın Şərq və Afrika arasında çölkü yaradan mühüm coğrafi bölgə olmasıdır. Bu araşdırmanı yapmamızın məqsədlərindən bazıları şöyle. Bu cür müşahidə olunan genetik müxtəlifliliyin nə vaxt və necə formalaşdığını bulmaq. Çöldə yaşayan insanların genetik soyunun nə cür və hardan gəldiyini öyrənmək. Qafqazda qən axışlarını, çöl imkanlarının istismarı gücləndiyi zamanda araşdırmaq. Avropanın çöl bölgənin və yaxın şərqdəki ən erkən şəhər mərkəzlərinin hansı vasitələrlə bağlandığını anlamaq. Bu səbəbdən 3000 illik bir dövrə əhatə edən 27 yerləşədən nümunələr Alman Arxeologiya İnstitutu və Rus həmkarların köməyi ilə toplandı. Xəritədə gördüyünüz kimi bu nümunələrin çoxu Şimali Qafqazdandır. Bu toplanan nümunələri təmiz otaq deyilən lablarda çalışırıq. Bunun üçün xüsusi paltarlar giyirik, çünki bizim DNT-lərimiz işlədiyimiz nümunələrə qarışa bilər. Bu diş ya da sümükləri təmiz otaqlarda toz halinə gətirib, onlardan DNT çıxartırıq. Çıxartılan DNT-lərdə topraqda tapılan mikrob DNT-ləri də mövcuddur. DNT yakalama adlı üsulla bunun içində bizim hədəflədiyimiz insan DNT-lərini alıb, təhlillərimizi bunun üzərinə yapırıq. Bu çalışmadan öyrəndiklərimiz şöyle. Qafqazın tarixindən əvvəlki 3000 illik dövrdə əhd edən zaman çəsişməsində ilk dəfə genetik olaraq araşdırdıq. Dağlarda və səmhər çöl rayonlarında yaşayan qruplar arasında genetik fərq olduğunu gördük. Bu qruplardakı genetik fərqlər ekocoğrafi bölgələrə qarşılıq gəlir. Görürük ki, Qafqaz dağları tarixdən əvvəlki dövrdə insanların hərəkətinə mane deyildi. Hər bir genetik klasterda zaman içində genetik davamlılıq və ikisi arasında arabir can axını var idi. Lola mədəniyyətindəki insanlar qədim Şimali Avrasiya soylarının ən gənc və ən qərbdəki mirasçısıdır. Yamyayadan başlayaraq çöl klastrındakı qruplarla əkinçilərdən gen axını gözləyirik. Mikroyərli zaman kəsişmələri olaraq kurqanlar zaman içində genetik soyların değişən hududlarını göstərir. Bir də digər tədqiqatdan öyrənirik ki, Qərbi Anadolu, Levant və bugünkü İran bölgələrindən olan fərdlər Neolit dövründə genetik olaraq fərqlidirlər. Yəni, yerel ölçəkdə genetik davamlılıq var idi. Son Neolitdən başlayaraq Şərq və Qərb arasında qarışma qradiyanı müşahidə edilir. Bunun miladdan öncə 6500 illər ətrafında başladığı təxmin edilir. Mis dövründən başlayaraq daha geniş miqyaslı siyasi dönüşümlərə rağmen və ya onlar yüzündən nisbi genetik davamlılıq müşahidə edirik. Çox güman ki, erkən Tunç dövründə Şimal Levantdakı gen axını Orta Şərqi Anadoludan deyil də Mesopotamiya və yaxud qədim İrandan gəlir. Və nəhayət, Alalaqdakı quyuda tapılan xanım uzun məsafəli fərdi hərəkətliliyin bir dəlilidir. Sağ olun. Thank you.
Thank you very much, Aysin, for your summary. Thank you so much, Wolfgang, for this very exciting and extremely interesting paper, and I would like to try to involve also the audience and ask